Quality by design isn't just regulatory compliance, it's your competitive advantage. Companies using these approaches achieve predictable manufacturing, reduce development timelines, lower cost of goods, and regulatory confidence that accelerates approvals. But most importantly, they deliver medicines to patients faster and more reliably. So the question isn't whether you need quality by design. The question is whether you will implement it before your competitors do. Welcome back to the Smart Biotech Scientist podcast. I'm David Brühlmann, your host. And in the last episode, we covered the foundation crisis that kills 40% of biotherapeutic programs during clinical development. Companies that never properly defined what quality meant at commercial scale. Today, I'm going to show you exactly how to build the process side of quality by design to prevent those failures. By the end of this episode, you will have the roadmap to go from manufacturing chaos to regulatory approval in record time. Here's the scenario that keeps biotech CEOs awake at night. Your process works perfectly at 2-liter bench scale. You're confident, investors are excited, everything is on track. Then you hit your first 50-liter or 100-liter pilot run. Suddenly, pH profiles look different. Titer drops by 20%, and you start troubleshooting, making adjustments, and trying again. The 200-liter production run brings new surprises. Aggregation appears out of nowhere. Charge variant patterns shift. And then your management team asks the question you dread. How long will it take to fix this? Your honest answer? Well, we don't really know. Could be 3 months, could be 12. Why does this keep happening to a lot of people in our industry? The physics change, mass transfer, mixing efficiency, heat dissipation, everything behaves differently at scale. The biology changes too. The cell stress might change, the shear sensitivity, the oxygen limitations create new challenges. Even the chemistry changes with buffer gradients and temperature variations you never saw coming. Most companies make a critical mistake. They confuse specifications with control. They focus only on the final product testing, like trying to control your car by only looking at the destination. By the time you see a problem in your final product, it's too late to fix it. What's missing is real-time process understanding, predictive capability, and risk mitigation that catches problems before they happen. But I'm already getting ahead of myself. We're gonna unpack all this in today's episode. So let's quickly recap the quality by design framework in case you haven't tuned in into part 1. And I highly encourage you to do so to get the foundations. So the quality by design framework we established in our last episode starts with your quality target product profile, or in other words, your QTPP, which defines exactly what you're building based on patient needs. From there, we identify your critical quality attributes, your CQAs, the specific product characteristics that directly impact patient safety and efficacy. Today, we are focusing on the third pillar of the QBD framework, how to control your manufacturing process to consistently deliver those CQAs. Not all process variables are created equal. The Pareto principle applies perfectly to bioprocessing. 80% of your quality issues come from 20% of your process variables. Quality by design helps you find that critical 20%. These are your critical process parameters, or CPPs. The ICH Q8 guideline defines CPPs as process parameters whose variability has an impact on a critical quality attribute and therefore should be monitored or controlled to ensure the process produces the desired quality. Think of CPPs as the vital signs of your manufacturing process. Just like monitoring heart rate and blood pressure tells you about human health, monitoring CPPs tells you about process health. Let me share a well-documented industry success story to illustrate that. Genentech's development of their monoclonal antibody platform faced exactly this challenge, a mysterious batch-to-batch variability that threatened their entire program. Through systematic QBD implementation, they discovered that small variations in feeding strategies during cell culture had dramatic impacts on product quality consistency. What was their solution? They moved from fixed feeding schedules to metabolic state-based feeding and control. So they were monitoring glucose consumption rates and lactate production in real time. The result was a significant improvement in process consistency, and this became the foundation for their platform approach that's now used across their entire portfolio. CPPs typically fall into two categories. On the one hand, we have upstream parameters, and they include pH, set point and control ranges, the dissolved oxygen levels, feed rate and timing, and also the temperature profiles. On the other hand, in the downstream processing, we look at the column loading density, the buffer pH and conductivity, the flow rates, and the residence times plus pool criteria and hold times. Now, let's explore the control strategy. In other words, your manufacturing GPS that guides your entire production process from raw materials to the final product. Like in a GPS system, your destination is your CQAs, where you need to be quality-wise. Your route is your process steps, so how to get there. And real-time guidance comes from CPP monitoring, which helps you stay on track. And when things go off course, you have process adjustments to recalculate your path. So the control strategy has three integrated layers. It starts with input controls. They cover raw material specifications, cell bank characterization, and medium buffer quality standards. We have process controls. They include the real-time CPP monitoring, the in-process testing, the release criteria, plus environmental monitoring, for instance, for burden and endotoxin. And then we have output controls. They encompass product release specifications, the stability monitoring programs, and innovative approaches like real-time release testing. Real-time release testing represents a major innovation. Instead of waiting 14 days for traditional release or even longer testing, companies can now release product based on validated process data and pretty much in real time. Obviously, if you look at it, it's much more complex than that, but we are very close to that. Amgen, for instance, has publicly shared how their implementation of real-time release testing achieved faster lot release and a cost reduction across multiple products in their biosimilar portfolio. The key insight is moving from reactive to predictive control. And traditional approaches detect problems after they occur, but smart control strategies prevent the problems before they happen. So in other words, you make sure that your critical process parameters stay within your ranges and they don't even go out. You know exactly what to do if it slightly deviates to actually keep them within these ranges. Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. So, while quality by design provides the overarching framework for systematic pharmaceutical development, one of its most powerful implementation tools is the design of experiments, or DOE methodology, which enables scientists to efficiently explore and optimize complex manufacturing processes. You might be familiar with traditional design of experiment approaches, but they have serious limitations because usually they're done in a one factor at a time testing, and it's slow and misses critical interactions. Full factorial designs test everything but require hundreds of experiments. And what is the problem? Well, it's a complex optimization, and it takes 2+ years using traditional methods usually to test whatever you would like to do. So you have to do some compromises. But there's a revolution happening in bioprocess development. It's called hybrid modeling, and it's transforming how smart companies approach process optimization. Hybrid modeling combines your existing biological knowledge with machine learning. Instead of treating your process like a black box, as it's done in traditional DOE approaches, it uses your understanding of cell biology, biochemistry, and engineering as a foundation. Then, machine learning fills in the gaps and finds hidden patterns you'd never discover through traditional experiments. The results are remarkable. For instance, DataHow, a leading process analytics company and modeling company, has demonstrated 60 to 80% reduction in required experiments across multiple pharmaceutical clients. Instead of 2 years and 200 experiments, companies are achieving better optimization in 4 months with 25 strategic experiments. So here is how it works in practice. Take CHO cell culture, for instance, as an example. Traditional approaches test pH, temperature, dissolved oxygen, and feed rates one at a time, then try combinations. But hybrid modeling starts with your knowledge of cell metabolism. Or in other words, it starts with all the runs, all the data you have done already. Even if you have done them with a different molecule, you can use that to build your hybrid models. So hybrid modeling starts with your knowledge of cell metabolism, then uses machine learning to optimize the interactions iteratively. Each experiment makes the model smarter, guiding you toward optimal conditions faster than ever before. For detailed case studies and published validation of these results, check out our previous episode with DataHouse founder Fabian Feidel and Michael Sokolov, where we dive deep into the science behind these impressive numbers. And the business impact is transformative, especially for smaller companies where every experiment counts. Months instead of years for development, and you'll be able to conduct fewer experiments, and this means lower costs and faster decisions. But most importantly, you discover optimal conditions you'd never test using traditional approaches because the model identifies non-obvious parameter combinations. And I recently had a conversation with Biotech engineer about DOE as they were preparing for their phase 3 studies. And they wanted to do traditional studies, but I recommended them not to do that. Instead, even if it comes a bit with a higher price tag, and that's free advice I'm giving you now, the ROI with hybrid model is much, much higher, which justifies also a bit a higher price tag to hybrid modeling approaches. So how do you build your control strategy? Well, your control strategy transforms process understanding into an operational reality. So think of it as translating scientific insights into manufacturing instructions that deliver consistent quality every time. Effective control strategies specify exactly which parameters to monitor, what ranges are acceptable, how to respond when parameters drift, and when to investigate versus when to take immediate action. They include decision trees that eliminate guesswork from manufacturing operations. So the most sophisticated companies are implementing model-based control. They use real-time data feeds into process models that predict quality outcomes before final testing. This enables a proactive adjustment rather than a reactive response. So consider feed control in cell culture. Traditional approaches use fixed schedules, but a model-based approach monitors the glucose consumption, the lactate production, as for instance Genentech did, and the cell density to calculate optimal feed timing and composition in real time. And what is the difference? Well, you'll get a much more predictable performance instead of a batch-to-batch variability. And if for whatever reason something changes, since you have a better process understanding and you have a model that works well, you can also make slight adjustments without deviating or going outside of your validated ranges. Training is crucial for success. And so you will need to understand not just what to do, but why you are doing it. So when everyone understands the underlying science, you will make better decisions when unexpected situations arise. Now, I want to give you a very practical exercise to do. Here is what you can do before your next meeting. Take one sheet of paper and map your current process from cell culture to the final product. At each step, ask two critical questions. First, what could go wrong here that impacts product quality? And second, will this step work the same way at 200 liters as it does at 2 liters? Circle the 3 most critical steps, and these become your focus areas for control. So this exercise reveals your biggest risks immediately and identify scale-up vulnerabilities before they bite you. The steps you circle become your foundation for CPP identification and the control strategy development. It takes a few minutes, but it could save you millions. So what's the bottom line? Well, every major biotech company that succeeded at scale has mastered these quality by design fundamentals. Quality by design isn't just regulatory compliance, it's your competitive advantage. Companies using these approaches achieve predictable manufacturing, reduce development timelines, lower cost of goods, and regulatory confidence that accelerates approvals. But most importantly, they deliver medicines to patients faster and more reliably. So the question isn't whether you need quality by design. The question is whether you will implement it before your competitors do. So remember what we covered today. Critical process parameters help you focus on what really drives quality. And then smart control strategies prevent problems rather than just detecting them. And on top of that, as a bonus, hybrid model can reduce your experimental burden by up to 80% while delivering better results. So here is your homework. Complete that 5-minute process assessment I've just explained you. Map your process, identify your risks, circle your critical steps. That simple exercise gives you the foundation for everything else. Thank you very much for tuning in today. If you haven't listened to part 1 where we talk about the quality target product profile and the critical quality attributes, do so. And please leave us a review on Apple Podcast or whatever platform you found us on. I love hearing from you. Tell us what you think of Quality by Design, how you like this episode, and thank you so much for taking this time. And I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.